Thanks for joining us again. As we sort of created this webinar, one of the things we really wanted to do was represent two sides of the spectrum. We wanted to represent the clinical side of the spectrum with a lot of the physicians who are living this and using it with patients on a day-to-day basis. But we also realized that this space is so new and so changing that we also wanted to have a really good representation of the scientists who are creating this for it to be useful in the clinical practice. And so I'm excited to introduce Dr. Lasky-Su here, who I've met just honestly a couple months ago. We've only been talking for a couple months, but she's been extremely informative to me as we sort of go into this multi-omics approach, which we'll be discussing, and how that relates to a lot of these epigenetic markers. And so thanks so much, Dr. Lasky-Su, for joining us.
Thanks so much, Ryan. It's great to be here.
We're really excited for this one. I think that we've got some really interesting things planned. But for those people who don't know you, can you give a little bit of an introduction and your background?
I'm an associate professor at Harvard Medical School and Brigham and Women's Hospital. And I really have had a research career. I'm a PhD, not an MD, although I'm married to a functional medicine doctor. So I get a lot of feedback. We have a lot of dynamic discussions about functional medicine and how it applies to what I do. And it's actually created a really great interaction where we can be thinking about these things, from both the research perspective and the patient perspective. I started my career, as many people did, in genetics. A lot of people who are in this area now started in genetics in the late 90s and have been looking at genetic markers and predictors of disease to begin with. And then since then, the whole field has really exploded and have now looked at a lot of other multi-omic data in association with various disease outcomes.
And so we're going to be getting into this idea of multi-omics, but before, I know that a lot of your specialty has been in metabolomics. And so just for those who aren't familiar with that idea, can you explain what metabolomics is and why it's interesting?
So I am passionate about metabolomics. Metabolomics can be summarized as the small molecules in any biofluid. And the reason I think this is so important is going to be really crucial in translational medicine in the upcoming years is, one, because we actually already use it. When we take a biofluid and we're measuring small molecules, we're measuring everything from lipid profiles to blood sugar levels to short-chain fatty acids, long-chain fatty acids, to environmental exposures, good and bad, to nutritional intake. The other thing about metabolomics that's really crucial, especially when it comes to the concept of multi-omics, is metabolomics sits as the downstream product of a lot of other omics. So when we were younger in high school, we learned about the central dogma, DNA goes to RNA, goes to protein, goes to small molecules. Well, in the omics terminology, we could say that is, genetics, there's epigenetics in there somewhere, genomics, proteomics, metabolomics, and disease. So metabolomics is really interesting because while it reflects this downstream consequence of your genes, it also incorporates outside exposures, right? When we are measuring things like lipid profiles or blood sugar, it's measuring what we eat. It also measures in exogenous exposures like environmental toxins. Metabolites measure the amount of ibuprofen you take, the amount of medicine you take. So you can look at this global profile of, let's say, 1,000 small molecules, and you can very accurately assess the biological state of a person. Now, one thing that is really interesting about the metabolome, and I'll contrast this to a lot of the other omics, is that the metabolome sits in this place where it provides clues about past, present, and future, all simultaneously. And if you think about that, that's a really interesting triad, right? It provides clues about the past with either genetics or, the downstream byproducts of what you've done in terms of what you've eaten or what you've been exposed to. It provides things about your biological, physiological state at the moment. If you just ran, or I just took a drink of coffee, my caffeine metabolite is going to be in me for about 3 days. And that's important. But then it also, if you look at it globally, can give a lot of clues into your overall health profile, and maybe things that are happening physiologically and diseases that are developing prior to actual disease detection. So because of that, I think it's very, very crucial to consider this when looking at disease and when thinking about its role in, treating disease and preventing disease.
I think that's definitely an important point, because we talk a lot about, or have talked about in some of these webinars recorded previously, a lot of sort of the predictive value of these tests, so that you can have a preventive medicine approach where you're actually treating conditions before they actually arise. And we've been talking about with epigenetics, but it sounds like the metabolomics is great at doing that as well.
Exactly. And, I'll even give you an example that it really, I think, highlights sort of how interesting metabolomics can be. I was doing a study on pregnant women and asking this question: can the in utero environment of a pregnant mother predict or prevent future outcomes? And I was looking at this. I was looking at the metabolome at multiple times during pregnancy of the mother. And I came across a metabolite that I knew. It was called theophylline. And I was particularly interested in this study in things that would protect against asthma development. And the reason I knew theophylline was because theophylline is a medicine that's been used to treat asthma. Yet we're seeing this in the mothers. Now, the interesting thing about this particular example is I knew theophylline is an FDA-approved drug, but it's no longer on the market. So I thought to myself, how is it that all these pregnant mothers are having theophylline? And, these increased amounts of theophylline are associated with reductions in risk of asthma in their kids. So I looked it up, and I realized that theophylline is one of the primary derivatives of caffeine. And that actually, what's happening is these pregnant mothers who are drinking more caffeine are naturally having higher levels of theophylline, which is a known bronchodilator, and that effect is actually affecting their children. So it's things like that where when we look at metabolomics, we can really see direct ways we can translate this into the clinic. And because so many of these small molecules are actually, already endogenous in our blood, or are drugs that we know, or are environmental compounds that we know, the treatment becomes super easy. And to me, that's really exciting.
I love that example. I think that it goes to show you some of the insights that you can develop that just sort of come out of nowhere, and how useful it is in actually clinical practice. And so I love that insight. I know that going back to this idea of multi-omics, I think that people are generally, again, functional practitioners especially, are familiar with things like genetics, a little bit less familiar with the epigenetics, transcriptomics, and maybe that middle line between genomics and disease. From a metabolomic perspective, how often do you also look at all those other multi-omics approaches and incorporate them into your investigation?
And I think this is a great point, Ryan. And I'm excited that functional medicine doctors are considering these things, because I do think it's crucial. And, I think when this field started, we all started with genetics. And if we really think about it, when I look at my perspective 20 years ago, and the perspective of scientists looking at genetics predominantly, they thought this is going to answer all of the questions about disease. And what we realized through the years and through technology development is that the best way to look at diseases is, again, in this holistic perspective, just like a lot of integrative doctors do. But that holistic perspective includes multiple layers of data, and that each layer of data can provide unique information that may not be able to be gleaned from other facets of data. So while we have genetics, and that can tell us something about our underlying genetic blueprint, what that tells is distinct from epigenetics, which really has become the hallmark of aging. And that is distinct from something like the metabolome, which now can really summarize well all of these other exogenous exposures, whether they be good or bad, that is independent of either the genetics and the epigenetics. We also throw in there then the microbiome, right?
Absolutely.
But one can argue that while each one of these can give us facets of information, for many disease outcomes, it's the combination of all of these working together that is going to give us the most accurate profile. And depending on the disease outcome would depend on how much one may influence someone more than another. And even depending on the specific, your specific causal pathway to disease. So for example, breast cancer, if you're someone who has a BRCA gene, that's a huge predictor. If you don't have that BRCA gene, it could be other environmental exposures. It could be other things. So when we look at multiple layers of these omic data together, it provides a much more comprehensive view that can give us clues into what's happening pathophysiologically, often that one omic data type in and of itself may not be able to do.
Yeah, I think that that was perfectly well said. I think that, as we sort of shift our topic from multi-omics to maybe how the multi-omics can predict or affect aging, I know that people who have heard some of the beginning of this have heard about some of these composite biomarkers having the highest hazard ratios and the most predictive value. And so I think that echoes a lot of what you're saying in terms of how we should focus our investigation. But going back to sort of the aging approach to metabolomics, I know that there have been a couple metabolomics-based aging clocks. Are you pretty familiar with those?
Yeah, there's been several groups that have developed different metabolomic aging clocks. Yeah.
So I know that in some of those investigations, they've been looking at maybe even just a few metabolites. Do you think that there is going to be sort of a larger based metabolomics approach, which might be able to yield other aging markers in the metabolome?
And this is a really great point, Ryan. I think one of the things that we recognize, especially in the last 20 years, is all of the generation of all these omic data types have really evolved, right? The genetic data we could collect in 2003, I always quote this, it was $10 billion and like 10 years of research to generate one human genome sequence, and now it's a little spit of blood, of saliva, and hundreds of bucks, and you can generate that same thing. Similarly, when we look at the metabolome, that's one thing we have to think of. Really, the technology over the last 10 years for generating a comprehensive measure of a lot of the metabolites in blood has really evolved. And so while there are a lot of different metabolic clocks, aging clocks, that have been generated, one has to consider that what's the input, equals the output, right? So if you don't have enough input, the output really isn't going to be as accurate. And I think we're at a point right now where really the amount of metabolites that we could actually consider in generating these biological aging clocks using metabolomics is so much greater than what it used to be, that now what we could actually measure and develop can be much more precise. The other issue around this is similar, as I said, in the past, all these other omics, it used to be so cost prohibitive to generate metabolomics on larger groups of people that, this is another thing that's required when you're developing a predictive algorithm is large samples that are robust. And that really hasn't become readily available again until the last 5 to 10 years as well.
And I think that hopefully that'll be an area of investigation we might be able to do, is to tie some of these bigger epigenetic data sets to metabolomic values and see if they might be able to predict some metabolomic values or other types of disease. And so I think that this is really all very, very interesting. And whenever I would just want to sort of summarize it with a question that I feel like clinicians are going to ask a lot, which is, knowing that all these multi-omics should be combined in order to get the best and highest quality data for predictive algorithms. What are you excited about? What types of investigations are you excited to look into, both from an aging perspective as well as just a general metabolomics perspective? And how do you think this will impact the physicians who are currently interested in this and wanting to be on that next stage of diagnostic medicine?
When it comes to multi-omics and metabolomics and predictive medicine, I do think, one thing that is incredibly exciting is looking at this combination of the epigenetic clock with the metabolomic clock. Science has shown that really the methylome is the basis of biological aging, and we can very accurately predict that. But one also has to ask, again, with what I was referring to earlier, when you can get independent or slightly different types of data and information, and you combine it with something that's already very good, what else can it tell us? And I think that's really where the metabolome is and integrated with the methylome is really going to be important, because I think, again, it's going to be able to give us increased prediction in unique facets where we can then explore and study. So to me, I think that's incredibly interesting. One thing I do want to say about prediction, though, that I think it's important for clinicians to realize and understand, is that prediction and predictive ability does not always imply causation. Now, for the example of the methylome and the biological clock, that is not the case, because we've shown biologically that predictive algorithm and that the methylome is the basis for aging. But often in other predictive algorithms, there may be other correlates that are actually highly correlated with, let's say, age, but actually are not causing it. So I think, one of the things I'm excited about is when we have these predictive algorithms, looking at the predictors, but then also considering the causation, right? If we can add more to different biological clocks like grim age and causations of mortality, we can predict it, but can we really identify ways to alter it, right? To alter that end state, because at the end of the day, this is about health and prevention of, adverse outcomes, prevention of death. And so I think looking into that and asking those questions about causation from a multi-omic perspective of what can we do on all of these different omics is going to be super important. The other area I'll say that I'm particularly really excited about with the metabolome is something that I think is missing in a lot of the other omics, and that is the step to clinical translation. Now, functional medicine doctors have done a great job in utilizing omic data to help patients with, disease outcomes. But one of the struggles we have as researchers when it comes to a lot of omic data studies, and I'll use genetics as an example, we may be able to identify a gene and say it's associated with a disease. But the process between identifying that gene and even that causal variant and a treatment is a very long and arduous process. Absolutely. One of the amazing things about the metabolome is it's almost the exact opposite. Because the predominant number of metabolites the predominant number of drugs or treatments that we use already in clinical practice are metabolites. Sometimes our research and our findings are as simple as identifying a metabolite. We realize it's already being used for a different purpose, and we can actually just repurpose what's existing very quickly and have dramatic effects on health. So to me, that step that we can very quickly go from identification of omic influences to treatment is something that is really exciting, because it just means that we can improve health in a more expedient way.
I think that that's an interesting thought, and I want to just follow up with one question on that, which is that you obviously mentioned the central dogma of biochemistry, and that's one thing I often bring up in the world of peptides and proteins. But as we get further down the line, looking from starting with genomics and then going from epigenomics to transcriptomics, would you say that general association to shortening the pathway to clinical treatment is often related to how downstream it is from the initial genomics?
Exactly. And I think that's one of the great things about metabolomics, because when you look at that pathway, it is basically the end thing before disease. Sure. And actually, in some cases, by definition, it is the disease, right? A blood sugar level of 126 equals diabetes. And that blood sugar is a metabolite. And so the thing about that, as you're saying, the more downstream you get, right? The one thing that I think is also very important to understand about that, as you become more downstream, inherently, what's influencing it is the environment, right? And on every level. And so that's why a lot of the more recent omics, whether it's metabolomics or let's say metagenomics with the microbiome, the key thing that it has is it has the influence of the environment incorporated in the measure. Not only does it have the influence of the environment, but because of that, importantly, you can modify it. And that actually is one of the things that I think is an inherent struggle with underlying genetics, is because it's not modifiable unless you're using something like CRISPR. Treatments are so much more difficult. But when you have something that is this composite measure of preceding omics with the environment, often there are a lot of ways you can modify it to affect physiology and health.
And we might be stepping out on this question just a little bit, but I think that if anyone's sort of new to this topic, they might have a similar question that's going through my mind, which is that because it's so sort of closely related to phenotypic disease and the multi-omic approach and this linear sort of downstream pathway, I think that one of my big questions goes to, is it the most sensitive early prediction method? Because, if it's so closely related to disease, does that often limit its ability to be very, very predictive?
Does it limit its ability to be predictive?
I should say it's sensitivity. I would imagine that things that are earlier in the pathways, like genomics and epigenetics and transcriptomics, might be a little bit more sensitive to prediction than metabolomics, just because the line between some of the metabolic values and the phenotypic values might be a little bit hard to find sometimes.
And I actually think this is a crucial point. And one of the ways that we disentangle this, it's all about the concept of study design, right? If we are looking at something, the design at how we are studying it is going to be incredibly important in terms of disentangling, right? Even the fact that, like, if I have asthma right now, my pathophysiological state and my metabolites are going to reflect it. Sure. Disentangling the occurrence of, both prediction and actually having the disease. And the way we do that is really through, when we are doing different study designs, we carefully design it where we say, okay, let's say we want to predict something like arthritis. Can we actually design a study where we're looking at people who are asymptomatic, let's say 5 years before prediction, and look at their metabolome then, and follow them, and then see who has arthritis, who doesn't, and compare the two. So when you have good epidemiological study designs, you can disentangle those two. But I think it's really imperative to consider that. And I will contrast that this is different than prediction, in the sense that often if you do a predictive algorithm with someone at the state where they basically already have the disease, their prediction is going to be high, but it has no value, right? Really? So when we do prediction, in the case of metabolomics, that study design component is imperative to identifying the early metabolomic biomarkers for disease versus the biomarkers that are basically synchronous with disease.
So I think that in general is a good summation of really the things that are necessary to have the proper investigation, and then what insights they can yield if you do have the correct data. I think that I think that one of the things we have sort of teased out is that a single omics approach is probably helpful but not comprehensive to sort of a physiological state, and it's better to combine those. And you have to do that through really well-designed studies, which can tease out correlation and causation, and really hopefully have both predictive value and treatment value, where you can decide sort of which is the best way to go forward and prevent that outcome. And so, so I'm really, really excited to see where this goes. I think that it's awesome to hear some of the projects that you're working on. Maybe on a future podcast, we can dive into some of those, because they're always fascinating, and really, as much as we talk about them being benchtop approaches, so tangential to application in every regard. And even studies like the caffeine in pregnancy, I think are really, really interesting applications. And so thank you again so much. I hope to have you back on as we talk about some other projects relating to the metabolome and the methylome, and look forward to talking a little bit more.
Great chatting with you, Ryan. Thanks so much.